Welcome, everyone. Today, I have the pleasure of introducing my own supervisor of my master's, Dr. Hafid Soualhine. Dr. Soualhine holds a PhD on molecular mechanisms of drug resistance in mycobacterium tuberculosis from the Pasteur Institute, and did postdoctoral trainings at the Laval University in Quebec and at UBC on bacterial genomics and recombinant TB vaccines. His career is devoted to TB and mycobacterial disease, with special interest in drug resistance and molecular epidemiology. And as chief of the National Reference Center for Mycobacteriology at the National Microbiology Lab, just across the street here, he's leading TB OMICS implementation, technology transfer to provincial laboratories, and collaboration with international consortiums on innovative bioinformatics pipelines for TB. He's also managing mycobacterial research projects on drug resistance and TB surveillance using innovative omics technologies. So please join me in welcoming Dr. Soualhine.
Thank you, Morgan. So thank you for this symposium organizing to invite me today. So I have a great pleasure to give you some overview of what we are doing at NML on TB, and also about TB surveillance nationally. So first, I want just for territorial acknowledgement, I acknowledge that as federal employee, as professor at the University of Manitoba, I work and live on the campus that are located on original lands of Anishinaabe, Cree, Orijji-Cree, Dakota, and Innu people, and the ancestral homeland of the Métis nations. So I respect the spirits and intents of treaties and treaty making, and remain committed to working in partnership with First Nations, Inuit and Métis people in the spirit of truth, reconciliation, and collaboration. So I have no conflicts of interest to declare. All my projects are funded by the government, federal governments or provincial governments, and some collaboration with academia. So if I see some companies or suppliers, it's out of my attention. So first of all, on the laboratory perspective, so Mycobacterium genus, so all we are talking about Mycobacterium tuberculosis, but there are many other mycobacteria that we're always missing, so that are non-tuberculous mycobacteria. So the genus of Mycobacteria since its discovery in 1886, so there is a delay in mycobacteria research for more than 1 century. So few species were discovered in the first 80 years, but still late 90s, so it was only 84 species, and now we are about 195 validated species of mycobacteria. So all we think about Mycobacterium tuberculosis. So even within the complex, there are other species that are involved, and we pay less attention to the species. And of course, non-tuberculous mycobacteria, mainly for rapid, slow-growing or rapid or slow-growing mycobacteria that are very interested and making some trouble for the public health sector. So I will talk sometime about Mycobacterium avium complex or Mycobacterium abscessus that they are really concerning. So for Mycobacterium, just it's well known who can see for the first time a colony of Mycobacterium tuberculosis. So about the TB incidence over in Canada, so my colleague Pierre, he already highlighted lots of information, so I will be brief on it. So roughly during the last 2 decades, the incidence of TB in Canada was stable, and on average it was between 4.3 to 5.2% per 100,000. So it's stable. If we see in detail during the subgroup of this slide is shown for the HIV and TB infection. So as you can see here, we have a high percentage of HIV reporting cases, but since 2017 and 2020, we see a little bit higher case of the status of HIV, but the number of TB and HIV co-infection is decreasing. But during the COVID, we see slightly few cases that are again increasing in 2020 and 21. So when we see the TBD size by population subgroup, so I will only focus on the Inuit population here, that we see some increase of the incidence, mainly in the last decade for Inuits. So we think that for the incidence of TB in Inuit were decreasing at the end of 2020, but unfortunately it was decreased during the COVID, and due to some access for the health diagnosis and reporting case, we can see here in 2021 that we have an increase of case in 2021, and it's the same scenario in 2022. So for the first nation, the incidence was stable, and it was really slightly decreased. But for the Inuit population, it's really concerning about this high incidence during the COVID now. So I want to highlight only during the last decade in the Inuits. So we see some case in some community in northern Nunavut or Nunavut, and even in Manitoba, the incidence was very high in some specific communities. So in our agency, we pay some attention to these communities, and we did some intervention that was funded by the Public Health Agency of Canada. So we have the NML team was trained on the TB deployments in the north, and with the collaboration with Nunavut government and Quebec, and also some cases in North Saskatchewan, we have deployed our team mainly in Nunavut. And you can see one example of our team that were deployed in Cape Dorset, and all the operation, they are managed from our emergency operations center. So we have the NML group that are deployed in the northern communities. They are well trained. They are competent about wide population screening for active and latent TB. So for the active TB, we do the screening using the molecular rapid testing with GenExpert. So as you know, for Mycobacterium tuberculosis, the classical diagnostics is very time consuming, and with the molecular method, we can detect tuberculosis on site in 45 minutes. In parallel, we can screen for latent TB using the IGRA testing, and for more than 5 years, this approach make really very good results in some communities, and we can see some decrease in the TB incidence in that community. One other point that we focus on this operation is about capacity building in the north, and we have organized some training for some technical staff alongside with our staff at NML and also on sites. And now the vision that we have is for some self-community testing in their own communities by deploying some molecular machine as points of care testing. So we hope that we can perform some integrative diagnosis, including HIV, TB, STBBI, and other infection with the same points of care testing. It's very challenging, so I thank my team that are always ready to deploy in very extreme cold temperature in the north. And for now, we are planning hopefully that we can plan some other testing in the north. So for the Inuit communities, lately during the pandemic, we have some outbreak TB that were ongoing, mainly in North Saskatchewan, in North Alberta, and now it's in North Ontario. We have very recent one in Pond Inlet in Nunavut, and in Panneertuk that were ongoing during the COVID. The problem that we heard about Nunavut, and we are discussing with some partner, is during the COVID, so the TB diagnosis was missed. In some cases, they were just halted, and you can imagine that some people from Panneertuk that need to travel to Inuktitut for access TB testing, they were just with COVID canceled, and they cannot travel. Or maybe in Nunavut, that for example in Red River, George River, or Kinjwaluswak that need to travel to Kujwak, And we have some positive cases that are in these hamlets and unable to travel for access to TB. Or even if we have some planning travel between these two villages, you can see that one positive case that's go till Equalute, and there is no diagnosis there. So it's really positive, TB positive. There is no treatment, no diagnosis for sure. So they have to send their sputum in the south till Montreal, and they have to wait for 5 to 7 days for just smear test. And they came back to their own village within other 10 or 15 people. So they are coughing, they are positive, no treatments, and then when they receive the smear positive or gen expert positive, they have to fly back another flight with another 20% for transmitting. So you can see that it was some disheartening story during the COVID or before the COVID for this access. It's why that we think this point-of-care testing is extremely helpful for TB surveillance and control in the northern communities. So this year, we are discussing with Nunavut to deploy in Penjint Rock and Pond Inlets. And even during the COVID, we support them with our laboratory-based expertise and also with genotyping, by genotyping all isolates from these communities experiencing with a TB outbreak. So about drug resistance, so for AMR prediction, so you know that there is phenotypic, the classical phenotypic testing, and now we are moving to the molecular testing. There are many efforts that were done about molecular sequencing or just point-of-care testing, the rapid testing. But now I will be more talking about the genomics and whole genome sequencing. That is a really very important technology to implement in mainly industrialized country, and we hope that one day we deploy point-of-care testing based on the whole genome sequencing. So I will be brief on the just classical technique, but for TB there are 4 main drugs. So isoniazid, rifampin, ethambutol, and pyrazinamide. And in Canada, we were testing for a long time these drugs based on some fluorometric methods using the proportion method. And since 2011, so we are testing the second-line drugs, all the injectable, quinolone, and others. So we have a good surveillance for drug resistance in Canada for many years, and maybe you know that TB surveillance program is the oldest one in Canada. So we have huge information that's asked for TB trend in Canada, including the drug resistance. So this is one example of drug resistance report that we are collaborating with our colleague in CCDIC. So every year we reproduce this classical report based on the number of cases, number of drug resistance, mainly the first line, isoniazid, rifampin. We calculate the multi-drug resistance, how many extensive drug resistance. This is the classical report to show the trend in. And roughly during the last 10 years, we have stable drug resistance percentage about for any drugs. So we have very low case of extremely drug resistance. We have roughly some MDR that are concerning, but they are under control. But for the trend of the resistance or the rate is around 10% roughly for any drugs. But the maximum, the monoresistance, that's higher, and we can see that the monoresistance is related to isoniazid or pyrazinamide resistance in Canada. For multi-drug resistance, most of the cases they are imported, so due to immigration, and we know that this is very challenging. So it's why in our lab we are very interested to study this drug resistance. So classically, the drug resistance is known as resistance to isoniazid, rifampin. But during the COVID, WHO released new definition of extremely drug resistance. This is the classical one, amikacin, capreomycin, kanamycin, and quinolone for XDR. But now there is introduction of pre-XDR isolates, including rifampin-resistant tuberculosis and 1-fluoroquinolone. And the extremely drug resistance case, they are determined as MDR plus resistance to 1 quinolone and 1 group A drugs, including bedaquiline levofloxacin, moxi. So if we see this new definition, NML, or in Canada, we have already most of the drugs that were tested, but we are missing bedaquiline testing in our lab. So all the academia and public health stakeholders, they urge us to implement this new testing in our lab. And there is another one regimen that include pretomanid and dilamanid that is really recommended for extremely drug resistance or multi-drug resistance, but we cannot access it because they are in their license. So Mike Stobart was leading this work on new drug developments at NML. So we designed new microplates using bedaquiline, clofazimine, and you can see levofloxacin and other drugs. But this new testing is based on micro-dilution, so we can determine exactly the minimal inhibitory concentration, the level of resistance, and we can also use molecular testing to predict the drug resistance to these all drugs. So the TB drugs for TB using whole genome sequencing prediction. So why we are interested in, first we need to answer some surveillance question. We need to understand how this mutation evolve in Canada and how to predict the right resistance to any drug in the future. So one big question that people are every time asking, can whole genome sequencing replace the phenotypical testing or the culture? This is one question that I don't have the answer now, but maybe it will happen. And another one that we really will take attention to this, can we one day deploy a point of care testing based on whole genome sequencing in remote communities? So this is what we are working on. So regarding some molecular improvements for TB, so here, for example, I can show you, maybe my friend, they are laughing about the old school. So when we did some sequencing, we have to amplify, we have to design our primer in-house, so we can synthesize even our primers and do the lots of amplification and the electrophoretic jets for sequencing. So we run a big gel between two glasses that is very thin based on area. We use the radioactive, the S35, and imagine that you're working for one week and finally you broke your gel, so we have to repeat this again. So it's time consuming, it's expensive, but now you have access to this very smart technology, so you can just run your culture, extract your DNA, submit to the DNA core, and you have lots of information in 48 hours. So it's, and also for when this is the third generation sequencing, including MySeq from Illumina or Me9 or Gridanian from Nanopore technology, and Ion Torrent too. So this technology, I remember when I started my de novo sequencing with high throughput sequencing, just for sequencing one box, that's Onococcus only, it cost us around $34,000 for just one single culture. And so it was extremely, and it took us more than 7 months to get 64 contacts. You are laughing, I know. So now the cost is really decreasing, so you can do some benign with that costs you $115, and you have a lot of information. So this is why the question where we will making this big data for sequencing, so, and how we can exploit it in the future. So this is an example of a nice output from your bioinformatic analysis, and you have all this mutation in maybe 48 hours, and you can have lots of information on drug resistance, about the cluster, about the genotyping, which lineage, and the different mutation in different targets regarding first line, second line, and new drugs too. So we validate this method at NML, and we choose Manitoba as pilot project. So we work first with closely with Manitoba isolates, and now we have this bioinformatic pipeline with the supports of our bioinformatics group at NML. So this is BioHansel for identification. This is NML tools that were built. Now it's updated with Genevieve and Rashid. And also for the micro predictor that can predict drug resistance for many drugs. So finally, it's compatible with our informatic laboratory systems. So we can send directly the report to our clients for TB treatments, for the TB management, TB patient management, and also report this mutation to the public health. So during the 2018, so we did this table is a little bit old. It's about 700 isolates, and now this validation is ongoing under lead of Minu Sharma and Mike. And we have high sensitivity for isoniazid and Rifampin, but you can see that we have really low sensitivity for ethambutol and pyrazinamide. This sensitivity that is low, it's really so sometimes we have it's really hard to associate some mutation with drug resistance sometimes, and you can find this mutation in resistant strain, and also they are found in sensitive strains. So the interpretation is not clear. It's why we talk about low confidence mutation or high confidence mutation. And sometimes we have some discrepant mutation that we can find in most of phenotypically drug resistance, or even if you have some phenotypic resistance, but there is no mutation in your targets. So we have a lot of question about the accuracy and specificity, sensitivity for this whole genome sequencing interpretation. So the same thing happened with the second drug resistance that we are validating at NML, but roughly we have a good specificity for the second drug, and we are also working to validate the same drug prediction for the new drug, mainly for beta-quilin and delamanid. One challenge that we have for whole genome sequencing for OMICS technology, so this is a continuous updates of our tools, mainly for identification, drug resistance prediction, to look for any mixed culture or contaminated one, how to eliminate sometimes the human DNA with some primary samples, and also for genotyping tools or for TB surveillance. We have some tools that we need to adapt for easy national TB surveillance in Canada. The one that we are using is SNPfield at NML, so it's SNPs-based tools, but we are evaluating others programs like GDI and CGMLST for tuberculosis genotyping. One thing that is challenging for TB genotyping, so when we implement the old method using the repetitive elements in Canada, so we need to send an easy interpretable report to public health nurse, and we are focusing on the same thing for the whole genome sequencing tool. So it's not easy to implement in the national level, so we need to engage public health stakeholders. They need to be trained. We need to give easy way to reports for the public health, for the contact tracing, the outbreak investigation. One example of the classical method based on repetitive elements. So it's based on some genetic markers in the chromosome of tuberculosis complex. And the known one is based on insertion sequence 1621 for RFLP. And the one that was used since 2008 in Canada, it's based on miroventer that are repetitive elements in the chromosome. And we send an easy report based on 24 digits, so it's easy to interpret. If you have the same single number digits, so that means your strands are related. And this work was led by Minu Sharma on the miroventer genotyping in Canada. So you can see how that we were receiving some isolates from different provinces, and we have more than 7,000 isolates in our database. So we can track each cluster that was circulating in the province. Here, an example of the genetic cluster that's were in Manitoba. And we can see some spike and decreasing of over time. For the cluster, we have in Canada mainly the cluster 2327, 1166, and 2742 that they were preponderant in Canada. And the same scenario for other provinces, and you can see that this cluster is unique for Manitoba, but very few cases they were detected in another province, Saskatchewan or Alberta. For whole genome sequencing, so we are using the SNP field for SNPs-based genotyping. So we look for similarity and difference between the sequence file that we have. We look for SNPs, some points mutation and difference. One thing that is important is about we need to have high quality of your sequence. We need to cover all the maximum of your genome and to validate every position that's analyzed between the difference. And when we have the tree and we have the result upon analysis, so there are some interpretation depending on the setting of your TB transmission, if it's on urban cities or in northern communities, and that we have to set up the cutoff value for SNPs and determine if they are really related or not. One example that we have done in North Quebec, in Nunavik, in collaboration with the McGill Group. So this was an ongoing outbreaks in from 2011 to 2013. And in one George River communities, it was around 900 population. And you can see that we had in that time around 90 people with TB. So that means it represents 10% of the population were infected with TB. So when we collect all the Mycobacterium isolate in this population, so we have about 50 cultures that were positive. 48 of this population, they have exactly the same mirror patterns. That means the same 24-digit number. So it was a nightmare for contact tracing and to do the epic connection for the transmission in that community. But when we did the whole genome sequencing, that is more resolutive. So you can see all the dots in red here. So we have three subtypes of the circulating strand, and this is A, B, and C. So the number of SNPs difference between B and C was only 2 SNPs, and the difference between B, C, and A it was 8 SNPs. So they were very close, and we have a clonal transmission during the community. So using the classical mirror winter, you cannot resolve the transmission. So this is a nice example of how the whole genome sequencing can resolve transmission dynamic of TB infection in one clonal community. So the setting is different when you are analyzing this transmission or contact tracing in Montreal or Winnipeg. So we have diversity in your clone, and the cutoff can be between 15 to 25 SNPs. So just a good interpretation of your results is depend on the quality and also on the setting when you are working with. So here, an example of one SNPs analysis based on few case isolates that we sequenced. We have here one isolate that's with the bad quality of sequencing. So you can see the interpretation that the SNPs are very close, and this is false interpretation. So when we remove this, and we have high good core genomes, so you can see that all the same isolate, they are really distant from the reference or between each other. So after the validation of this drug resistance and working with the TB genomics, so we have clear vision to share this whole genome sequencing with most of the provinces in Canada. So when the proof of concept system is validated, so our intention is to share it with our provincial partners and to be able that they use their own whole genome sequencing. So we have collaboration with the province in green. So we share the whole genome sequencing with them. So they will have very soon the access to the bioinformatic tools, and We have close collaboration with our colleague in Ottawa for drug resistance prediction. Our hope is to report this year some molecular WGS-based surveillance in Canada using the new drugs and also the first and second line drugs with our colleague. And in the near future, we are hoping to implement the whole genome sequencing with our collaborator in Canada. Maybe in 2 or 3 years plan, short-term plan, we have to work closely with our partners for TB surveillance using different methods. Even with SNP, we think that SNP field analysis, it's hard to interpret by public health, so we are looking for other alternatives in the future. So for whole genome sequencing transfer, some challenge that we have is to get our partner access to this information. Some data sharing agreements between different jurisdictional partners. So we have to deal with every information with different provinces, and the policy is different from one province to another. And also another big challenge is how to cooperate with the international consortium. So because they are different ones, so you have different mutation database, which one we need to select and which one we have to deal with, how we can interpret the whole genome sequence data, for example, with the Cryptic Consortium or the TB Profiler or the Australian Public Health organizations. So the output from this technology is, for now, it's very different. So when we have which one we need to select for our Canadian partners. So in the future, we have planned to work on machine learning that is very attractive for genotyping, mainly for TB surveillance. And we have some agreements and research agreements with the big consortium in Europe and United States. So from the last 4 or 5 years, we have in mind this challenge, the standardization and proficiency is a big challenge. How all our province partners will use exactly the same protocol, the same output, the same bioinformatic pipeline, and the ethics, which information we have to get access in. Should we share this information with others? So this is some agreement that we are facing now. And also some legal and sociopolitical concern about patient management treatments. And what is a question that raised me recently, what is really the benefits of whole genome sequencing or national TB surveillance, and what is the deal with some international partners? So how we can implement some surveillance or survey with importing some case of multi-drug resistance, for example, in Canada. So as I told in my introduction, lots of work focused on tuberculosis, but little is known about non-tuberculous mycobacteria. So this one nice example that we did, and Michel published this paper on abscessus. So when I was working in Quebec, so roughly we have between 8 to 9% of our clinical isolate that were tuberculosis complex. The same ratio was roughly for Mycobacterium abscessus, but one that is really concerning is Mycobacterium avium complex. It represents between 38 to 43% of the isolates. So it's a huge number of MAC. And for example, for abscessus, there is ongoing outbreak in pediatric clinic that they have the median otitis in pediatric, and we isolate Mycobacterium abscessus in that case. So we have 3 cases that were very similar, and one case that was Mycobacterium macilliensis, different from the other one. A second example, it was a tattoo parlor that have 11 patients having no ATM infection on the skin. So people who love tattoo, be careful that you go in very hygienic tattoo parlor and be sure that they are using sterile water. And it was not published, but I learned that from this tattoo parlor, the ink that was used was contaminated with Mycobacterium abscessus. So be sure that they are using sterile and hygienic condition for tattooing. So our plan, we are working on NTML for abscesses, and also we are using GMLST-based genotyping for abscesses, and also for drug resistance prediction for abscesses. And the same thing now is working with EMA about MAC genotyping using whole genome sequencing for drug resistance, and also for some other mobile elements that responsible for drug resistance in MAC. One item that we were really attracting to our team is how to use rapid sequencing using whole genome sequencing directly in sputum specimen. And this is very challenging technology. We were aware about the challenge that we have. And recently, Oxford Nanopore, they released their protocol that can be used for rapid drug resistance, including some for first-line and some second-line drugs. Most of the published paper, they work for whole genome sequencing on positive culture. But how about primary specimen like sputum? So the challenge, if you extract mycobacterial DNA, we know that mycobacteria, they have waxy cell wall. They are very hard to lyse, and very challenging to get high-quality nucleic acid extraction from them. So, and also from primary specimen or from tissue, the mycobacteria, they are intracellular. So how to lyse the eukaryotic cells and also mycobacteria, it gets a good quality of your nucleic acid. And when you are analyzing tissue or sputum, you have maybe less than 0.01% of mycobacterial DNA, but most of your nucleic acid, they are human, they are eukaryotic. So how to deplete all this huge amount of eukaryotic DNA and focus your analysis only on mycobacteria. So for most of whole genome sequencing directly from specimen like COVID, so you have small viruses that are around maybe 25 to 38 kilobase. So you can do some genome amplification using to pull some primers. It's easy to do, but for using a genome of 4.4 megabase, it's very challenging to amplify all the genome. There are other methods also that use the targeted gene or target sequence to amplify maybe less than 1% of your genome. So you can have a whole genome sequence data, but you miss 99% of your genome in your chromosome. So in our project, we did a balance between genomic, the chromosomal DNA, and also the targeted genome when we started this research project. So this was led by our student, Michelle Wozinski. So she starts, thank you for, she was really challenging. So we know that we are living in an area that is new to us. So the protocol that we developed, we was focusing that our protocol needs to be compatible with the hospital lab that we are using. So at the beginning, we decontaminate with the same protocol using with NaOH and N-acetylcysteine. So we did some osmotic lysis to get rid of cells, and then we deplete our eukaryotic DNA. We use simply the DNase 1, and then we extract the mycobacterial DNA that are resistant to NaOH and resistant to DNase here. Finally, when we got the mycobacterial DNA, so we keep the genomic DNA, even if it's little, but it's very important to our sequencing method. We did some multiplex targets, so to amplify the resistant targets for different genes. And we do randomly amplified genome using the Genome5 technology. So we pull all the amplified and genomic DNA, and we submit to Illumina sequencing and also for Nanopore sequencing. We have already the bioinformatics tool for culture, so we apply it for sputum. So the application of this method is for short, we hope that to get shorter around time for TB treatments to adapt the treatment for TB patient. Also apply this new OMICS method for TB surveillance, so we can do TB outbreak investigation or clustering TB in Canada or elsewhere. And we hope that we apply this point of care testing in northern communities in Canada. And for sure, there is many paths for the research, including fundamental and applied research on whole genome sequencing data. So Michel worked on different protocols to do for DNA treatment with and without, and we have some different pool for the genomic and for the amplified. We use some specific primer that are GC rich to amplify randomly some region for the genome, and you can see that the protocol with DNA treated, they give us much better depletion of human DNA. And we have the ratio of bacterial, and also another challenge that we have other bacteria in the flora that we need to eliminate. But for this case, for this discrepancies, it's a discrimination between bacteria and mycobacteria. It's really hard to achieve. So this is primary results for this sequencing outputs directly from sputum. And you can see that the genome with one coverage with adaptive sampling. So the adaptive sampling, this is another method to deplete or enrich mycobacterial sequencing. And with some samples, we can have greater coverage using the adaptive sampling. That means the eukaryotic DNA depletion works with this method. So this is comparison between Illumina sequencing and Nanopore. And to get the main coverage with Nanopore, you can see for some case we have greater coverage using Nanopores because we have a long read sequencing. And with Illumina, for some others, it's really the same output that we have between Illumina and Nanopore. For AMR prediction, using even spiked specimen with BCG or with clinical isolates. So this is primary results on 19 specimens that we analyzed, and we have 6 out of 19 that were failed, but we have 2 that give false results for drug resistance prediction, mainly for pyrazinamide and isoniazid. But the surprising results that for Nanopore, that is really now that it has some error for sequencing. We have no false positive results. And also the filer with Nanopore was less than Illumina. So these results, they are very encouraging on the output that we got from either Illumina or Nanopore sequencing directly from sputum specimen. When we compare our results with the published one, so we have very few cases that are published directly on sputum, and the coverage that we have is between 7 to 20x for the genome, and the sensitivity is very low about drug resistance detection directly in the sputum. So this is our protocol that we are working on. So we are collaborating with Manitoba and also McGill Group and BC CDC for this work, and we have further clinical specimen to assess with this technology. And also we are comparing the Nanopore company protocol with ours, and we hope that we can deploy this method in the future. There is another attractive method that we apply the same protocol using for lepromatosis, for leprosis. So we had one positive case for leprae, Mycobacterium leprae, that was positive by real-time PCR. So we were able to multiplex some primers for identification and also for Rifampin and Dapsone and quinolone resistance, and using for now the Illumina sequencing directly from the skin testing. So we were able to get a high number of reads, And by Sanger sequencing, also we identify this strand is. This is another venue of this whole genome sequencing that can be applied for other mycobacterial disease like leprae, or the new emergence of new variants from leprosis that is called Mycobacterium lepromatosis. So we are doing the same using Nanopore. So hoping that this protocol works also for leprosis, and for the Mycobacterium leprae is not cultivable, so it can be applied directly on some sputum. And we are looking for international collaboration to apply this method. In whole genome sequencing, as I say, low and high confidence mutation to interpret the drug resistance, it's not easy. It's really a deep knowledge of different mutation. And one example, for example, is ethambutol, that is a bacteriostatic antibiotic used for TB treatments. So Morgan is leading this work, and when we start this whole genome sequencing, what we look every time for some mutation in OMBB that is present, but the phenotypic test is always sensitive. So we ask ourselves, why is this discrepancy? When we look to compare with some published work using Sanger sequencing or other methods, so we found that this mutation is present in susceptible and also in resistant strands. So the predict PPV is low, and the sensitivity is really low. For our collection, so we collect all these resistant strands. So Morgan did the sequencing, and also she includes some control strands that are sensitive. So there is no mutation in susceptible strand, but there is mutation here. Phenotypically, they are sensitive, but the mutation are here. So there are two types of mutation. So the aspartate and alanine mutation in this codon, and for the critical concentration, that is the standard one at 5, all the strands show some sensitivities here, but they harbor this mutation. So the molecular prediction is resistance, but the phenotypic is different. This is not true for few cases, that there is some correlation between mutation and drug resistance, but this result show that, and when we tested the susceptibility testing using different concentration, 2, 3, 4, and 5, so we found that this mutation is associated with low level of ethambutol between 2 and 4, and few mutations that were associated with the resistance at 5. So you can see that discrepancy on phenotypic and whole genome sequencing data and how we interpret these discrepancies. So we think that some mutations are associated also with low level of drug resistance, and the minimal inhibitory concentration is a good way how to identify this association. We look beyond the MBB gene, so when we look into the operand, we found other mutations that maybe some exacerbates these patterns of low resistance to ethambutol. Some interesting mutation in the MBB and also for the regulator on the operants. So we found this frame shapes that is associated with this kind of mutated codon. For sure, there are other investigations at the fundamental level to look at what is the extent and what is the influence of this mutation. So just to show you an example of how to resolve some discrepancy interpretation between phenotypic and genotypic. And other groups, they are calling for changing the critical concentration of in vitro testing for ethambutol and even for other drugs for tuberculosis. One other challenge that we have also for pyrazinamide, we found a cluster in Manitoba that is the strand is circulating in one family, and it's sensitive, but in one year we found the same cluster harboring one isolate that is resistant to pyrazinamide. So when we look to the mutation, there is no mutation in the main gene that is PNCA genes. So we ask ourselves how this resistance evolved during one cluster in Manitoba. Some classical published papers to look for new mechanism. Normally, what they are doing, they are inducing resistance in vitro, but we have a very interesting case that will look for some parental strain or some paired strain in the same patient who harbor sensitive and they evolve to resistance during the treatments. But these cases are very hard to find. Another alternative is to look for some cluster circulating, for example, in one family or in one cluster, and there is a change from susceptible to resistant strains, and we are interested in it because we have another cluster in northern communities. When normally for northern communities, we are happy that we have most of them, they are susceptible, but one day you can see some evolvements of pyrazinamide resistance in Nunavut, and we have one cluster in Nunavik in North Quebec with isoniazide resistance, and we are very interested to study this mechanism, how this resistance evolve in one communities, because in northern communities, the transmission, as I say, is very clonal. So Morgan, she is working on this project, and she's analyzing the difference between the resistant strain and the parental sensitive one using genomics, and now she's developing transcriptomics and proteomics to look for this difference for the resistance. We are very for the other patterns. When we looked at genomics, we found some efflux pump with some SNPs in this pump, and we are sure that there are other mechanisms outside the classical targets for drug resistance. When we were working on whole genome sequencing identification during the last 5 years, so one for Mycobacterium tuberculosis complex, we know that the most one that we isolate is Mycobacterium tuberculosis. It's also some case of Mycobacterium africanum and bovis that we involved in zoonotic TB. In public health reports, unfortunately for the previous year, there is no information about this species, even if some provinces they are reporting the species identification, but during the last experience, I can say that Mycobacterium africanum, they are rare, between 2 to maximum 5 or 6 per year. And for bovis, it's around 2 to 7 or 8 cases per year. So they are, but for oranges, it takes more attention during the last 5 years, and some reports describe some cases that were described in South Asia. So when we were detecting the whole genome sequencing identification, we found that some SNPs based on GRAS-based genes that were also contained in Mycobacterium africanum and PNPD. So it was difficult to discriminate between africanum and PNPD. So we decided to do whole genome sequencing for all these isolates. And finally, Rashid was working on this project. And when we look to the literature that were published, there are some known genes that can differentiate Mycobacterium oryges from the other complex. And so we repatriate all the published genomes in the public database. Maximum, they were at that time 56 isolates described all over the world. And in Canada, we were surprised that we identified 61 isolates from oryges in Canada. Mainly, it was in 2 provinces, so in Alberta and BC case. So when we look to the SNPs-based whole genome sequence, so here for the GRAS-B, so only 4 that were known by the literature and included in our bioinformatic tools. But you can see in yellow and in red, those are unique for Mycobacterium oryges, and there are also some SNPs that are specific for other species within the complex. We are updating our BioHansel tools to discriminate between oryges and other stuff, and also we include some SNPs that are specific for other Mycobacterium species within the complex. When we look for the zoonotic TB due to Mycobacterium origins in Canada, so we compare what we are published in different countries, mainly and also for the animal and human Mycobacterium origins. So in the orange color, you can see all the animal isolates. Most of them, they are from India, but for human, so they are dispersed between India and Europe, and only in Canada, all these cases that we described, they are in green. So when we see the SNPs difference between the isolates in Canada, they are very diverse, but they are more related to some zoonotic TB that is imported from different countries. So surveillance for zoonotic TB is ongoing with public health in Alberta and also in BC to look how the zoonotic TB they are imported in Canada. And but if we see, fortunately, the treatments for the zoonotic TB they are susceptible to all drugs. In the preliminary results from Alberta, genotyping and surveillance in Alberta, all the cases they were imported from abroad, and there is no ongoing transmission in Canada within zoonotic TB. The same work is done on Mycobacterium bovis in collaboration with CFIE. So we are interested to look for the trend of zoonotic TB in Canada involving Mycobacterium origins and Mycobacterium bovis in Canada. And I would like to thank my team and all my collaborators in Alberta, also in Manitoba, and McGill universities, and also all the collaborators from BC CDCs, and would like to thank Dr. Denise, Natalie Knox, and Dr. Adams from University of Manitoba for their great support and collaboration. Thank you.